Okay, so today we are starting with the main genetics chapter that we have to cover as per your exam point of view. This is a very high weightage topic, one of the high weightage topic. So, principles of inheritance and variation is the foundational conceptual understanding how people finally got to know about how genes and different forms of genes are controlling all the life processes. So, when we want to see principles of inheritance and variation, three terminologies we must consider. Firstly, understanding of genetics. So, genetics is a branch of biology which deals with the various aspects of the inheritance. So, that will cover all kinds of a thing which will be inherited from the parents to their children, as well as the variation that children have. So, this is a complete branch that when we want to refer all this kind of a changes associated with it. Then the inheritance, as the word itself suggested, this is the process of carrying of the characteristic which can be passed from their parental generation to their children. That is the progeny that we use in the more or genetic word, the genetically inclined word is the progeny on the basis of the heredity. So heredity is a key of carrying out or passing out the characters throughout the various generations. Though we are saying there are characteristics which can pass from genetically inclined to the parents to their progeny, every generation have their own changes, their own characteristic with it, along with the one that have possessed by their parents also. So the degree of this variation is referred as a variation, by which the parents are different from their progeny, or moreover the progeny is different from their parents. So all these law or these statements are get visible proof based on the works by the Gregor Mendel, who is an Australian mathematician, scientist, worker, and he was referred as a founder of the modern genetics, the concept of the modern genetics. So his work based on this hybridization experiment, what he carried out with the garden pea plant for 7 years. And then, based on his observation, he proposed various law of the inheritance, how this living system is working. So, the main thing or the main advantage of the Mendel's work, the traits that he had selected and what kind of an experiment he has done. So, he shortlisted the 7 various characteristics of the garden tree plant. So, some of them were seen as a dominant, which can be seen in a majority of the population, and another is the word recessive, that is the this variation is based on the what kind of a combination of the genetic variations in them. So he selected 7 characteristics: seed shape, seed color, flower color, pod color, then the flower position and the stem height. Based on these 7 characteristics, he carried out various experiments. So what he did the artificial pollination experiment that we do right now. So selected one flowers, they remove their anthers or stamens, the process we called emasculation, and then they transferred the pollen from the any flower of the interest or plant of interest on there, and then check the progeny what it is referred to. So that is the main advantage of the Mendel's work. So for understanding what Mendel had stated during his law, we have to come across some of the important definition and some of the important concepts. Those are first important concept is the allele. So allele is a nothing but a genes which code for the pair of contrasting characteristics. So when though we are saying the contrasting characteristic, this is nothing but a slight variation from in form of the same gene, which can be a nucleotidely little bit different from the other. However, functionally, they can perform the same kind of a function. So if we are saying like that, because we already now aware of that we get the pair of chromosomes. So one we get from the father and one we get from the mother. Each chromosome will carry out the characteristic from both and mother and father. So the allelic combination that we get is based on we are getting from the both the parents. So two kinds of alleles can be based on their presence. We can divide chromosomes as a homozygous or heterozygous. When we say homozygous, word homo itself refers as the same. So it is the same allelic combination for the gene. So if we see in the diagram, Homozygous one is the, means say if gene is, we say B gene is there. So if same kind of a capital B kind of a combination will be the allele combination, it is homozygous. Also, if this is a small b and small b, which could be a recessive characteristic, but it is present in the same format, so they, at that time we called it as a homozygous kind of a chromosome. On the other hand, when there is a two variations present on a particular gene, is if they're on the chromosome, then we call it as a heterozygous one. So capital B and small b will be representing as a heterozygous combination. So these two concepts are quite important to understand what happens during the Mendel's work with the pea plant. Then two more terminology that we come across is the dominant factor and recessive factor. Dominant, as the word itself suggested, the dominant is something profoundly found in a particular expression. So here when we want to see in the genetic point of view dominant characteristic, this is a relationship between the observed trait and the inherited trait. So genetic makeup is whatever the genetic makeup it is, the observed characteristic that we are getting in a phenotypic way will be the one which is expressed more profoundly is what we referred as a dominant characteristic. On the other hand, recessive factor is the allelic combination which is always homozygous in nature. So both recessive kind of alleles must be present to express the recessive factor in the observational characteristic also. So that is the main thing that we must consider during this course of event. Then so few more important terminology is the what we referred when we do the Mendelian work is the filial generation or progeny. So filial generation or progeny is the one and the same thing. So this is nothing but the offsprings or children which are produced by the crossing over of the two individuals. So in the first generation, then we will define it as a F1 generation. If it is a second generation, we will define it as a F2 generation. Genotype is nothing but the genetic makeup or the combination of the alleles which is present on a particular locus. And the phenotype is a observable, means physical characteristic which we can see by our naked eyes. So that will be the appearance, development and behavior. That is how we are defining things. So Mendel used different characteristics, that the 7 characteristics that we have seen, and the main advantage that it has, he used the pure breeds to see the apparent change and then concluded his law of the inheritance. So say for example, one thing that he crossed one tall looking plant with the short looking one. When they are crossed, their next first generation is also tall in nature. So that is surprising for him. Then he crossed the two F1 generation plants and see what it gets. So he get at that time 3 tall plants and 1 dwarf plant, that is short plant. So at that time he got to see that there is some visible differentiation in it, which will be carrying out something that he referred as a factor, because at that time concept of a gene or understanding of genetic is quite limited. So at that time he referred the word factor, that is something is passing out from the gametes produced from the parental generation to their gametes, and then the new resultant is one which is appearing in a different way, whatever combination it has. So here the typical example is that P is referred as a tall. So alleles are always occur in a pair. So the homozygous tall plant when crossed with homozygous recessive plant, that is small t, the gametes will be identical. So that will be T T. So the resultant union will be heterozygous in nature, which will be capital T and small t. However, phenotypically, they are all tall in nature and genotypically, they are heterozygous in nature. So after that, we cross the F1 generation plant, that is the mating occur between the two heterozygous things. At that time, two different kinds of gametes will be produced. So one will be the for the capital T, another will be small t. So 4 combinations are possible. So by this particular combinations, we get a homozygous tall, then 2 heterozygous tall and 1 homozygous dwarf. So phenotypically means by appearance the ratio will be 3 is to 1 for tall to dwarf, and genotypically the ratio will be 1 is to 2 is to 1, that is 1 is pure homozygous tall and 2 are the heterozygous tall and 1 will be homozygous recessive. So when the Mendel observed these things, the word factor that he is referring to is actually the genes that we now understand what we want to say. So this is a monohybrid. Yes, yes, Shrey.
Ma'am, I had a doubt that
Mhm.
can one chromosome contain multiple alleles?
For the same gene, no. It can have the multiple alleles definitely, because this is one gene that you are referring to, right? Say for example here this is a complete chromosome. So each chromosome will have the means one chromosome will get from the mother, one allele you get from the father, right? So this is say for gene B. Say for another gene A, there will be a combination of the alleles will be there. It's like that. This is the same format with what we get from the different things, and by the combination what it has, it will be expressed differently. So each chromosome will have the different kinds of a genes with the different allelic combinations. By that way, yes, that is true. But for the same gene, only one allele will be there in each, which one get from the one father and another from the mother. Okay.
So, ma'am, so
Hmm.
only like one will be passed, right, from each.
Yes, yes, yes.
Okay, ma'am, got it.
Okay. So the most common method that we are using, it's called the Punnet's checkerboard method. So by putting it everything in a square and by making the combinations of the what are the possible resultants will be there. This is what we call the Punnet's checkerboard method, which is this one. So the same monohybrid cross that we have described. So monohybrid cross is nothing but the only we are considering only a single gene and see the what is effect in the different generations. That is what we call the monohybrid cross. So what Mendel had did the same thing, it's just put in a square. So it will be easy to visualize, easy to understand. So the tall, homozygous tall, heterozygous tall will be reduced. The one, only one type of a plant which will be tall in nature but heterozygous in nature. So to see it very clearly, this kind of a arrangement will help us to imagine things on a better way. So this square arrangement will make the possible combination that we have just seen right now. But this kind of a F1 progeny when crossed, we will get the 3 tall plants and 1 dwarf plant. Similarly, Mendel also checked whether whatever he is getting, it is true or not. So for this, we use the test cross. The word itself refers to where there is a one factor is unknown and another factor is known. So what he did, he chose the homozygous recessive one. So once he get the recessive is always pure in nature that we have already learned. So he choose the homozygous recessive kind of a flower. Say white color is a recessive characteristic and purple flower color is a dominant characteristic. So he choose one dominant characteristic by phenotype, one flower which is dominant in nature, purple in color, but we don't know whether it is homozygous or heterozygous. So for example, he did the crossing for that. So say for example, if the flower is homozygous dominant, then it will produce only one kind of a gamete, which will be capital W, which will be represented by this particular kind of a gene. And homozygous recessive is always in uh, identical alleles will be there. So that will be small w. So the result flowers are all violet. If this is the case, we know that definitely the unknown flower has the homozygous in nature. That's why all the flowers are produced by this crossing are violet in color. On the other hand, say if the flower is in the heterozygous dominant kind of a thing, so it will produce the two types of a gamete, capital W and small w. And at that time, we will get the mixed progeny, which will have the heterozygous dominant characteristic and homozygous recessive characteristic. So half flowers will be violet and half flowers will be white in color. So if this is the case, we can definitely know the unknown flower is heterozygous in nature by genetic conjugation, means its genotype is heterozygous in nature. So test cross will help us to find out the unknown genotype of the test, the question of that particular flower or particular characteristic is homozygous or heterozygous in nature. That way we can easily reveal what kind of a characteristic or the genotype associated with that particular phenotype. So based on this, yes, Shrey.
And so like we take the offspring and also
Mm-hmm.
then we take a homozygous recessive like, another flower like a
Yes,
homozygous
means
recessive.
at least
And
phenotypically we can say.
so can we take like instead of homozygous recessive like a heterozygous or homozygous dominant?
Homozygous dominant, yes, we can see that homozygous dominant can be also, at least you have to know genotype of the one character for sure. The thing is that just
Can
imagine,
also
just imagine if you take the homozygous dominant characteristic over here, so that will produce the same kind of a uh, same kind of a dominant gamete, right? That will be capital W. Okay. So if this is a dominant characteristic for the unknown, all the flowers will be produced are WW, which will be violet in color. Okay. Similarly, if this is a heterozygous one, and if you are taking the dominant characteristic, at that time also, if though this as the purple color is a dominant in nature, you will get this small combination also, but it will phenotypically also it will be producing the violet color. Then how will you understand what is the unknown in that, right?
yes, ma'am.
Yes, so that's
Mm,
why homozygous recessive characteristic will help you to understand that thing in a better way, to reveal the genotype of the
yes,
unknown.
ma'am.
Okay. So based
Okay,
on
ma'am.
this monohybrid characteristic that he has observed, he stated two laws of inheritance. So first law is the law of dominance. So law of dominance stated that the characteristics of the all the plants or the different factors are controlled by some of the units that he called as a factor at that time. Mendel referred it as a factor. And he also observed that factors are always occurred in a paired format. They are never alone, they are always occurred in a pair format. And in dissimilar part of a factor, one part is a dominant, one pair is a dominant, and another pair is a recessive. So one which is expressed profoundly, and one is occur when the pair is a homozygous in nature. So law of dominance will help to explain the various characteristic which is happening by the F1 generation in monohybrid cross, that is the only one character is expressed, and on the other hand, even for the F2 generation, the main dominant characteristic is present in a maximum population and recessive characteristic is present only a least population. That way, law of dominance can be expressed very easily. Second law that he stated is a law of segregation. So the word segregation is the differentiation, that we segregate few things in a different format. So this law is based on the appearance that though there are two different factors are present or in the pair format, they never mix with each other. They express independently. So in the F1 generation, say for example the case of the tall plant that we are referring to, in the F1 generation all plants are tall. At that time no dwarf plant is expressed. However, in F2 generation, again the dwarf plant is reproduced. So that means they are never mixed together. This segregation will help to reappear in the F2 generation. However, they never blend together. This is what he has observed as a law of segregation. So homozygous gametes, parent will produce only same type of gametes for the new progeny to have, and heterozygous ones are produces two different kinds of gametes which will have the equal proportion of the alleles. So that means the capital T and small t, these are the two different kinds of gametes which will be segregated somehow, because at that time the knowledge of the chromosomes and the genes was not profoundly found. Though these are the two main characteristics that we are observing, there are some of the phenomenon that we referred as a non-Mendelian inheritance. That means this kind of a phenomenon do not follow the Mendel's law of dominance or even for the segregation. First of such case is called a incomplete dominance. Incomplete name itself suggested that something is happening in a partial way. So this is also referred as a partial dominance. So in this kind of a cases, what happened? There are some kind of a in some plants or in some animals, the form of a gene interact with both alleles and eventually both are expressed in a partial way, resulting into the intermediate or different kind of a phenotypic arrangement. So for example, the evening rose kind of a flower is there. So in such flowers, if we can say there are red flowers and white flowers. So red flowers are dominant one and white flowers are recessive one. So when we do crossing for this particular flowers, we are expecting the F1 progeny should be, as this is R means capital R and small r will be produced, and that will be resulted into red color of the flower. However, F1 progeny for all these plants are pink in color. That means they are neither red nor white, but there is an intermediate kind of a expression of the phenotype is there. However, genotype is the what we are expecting. At that time, pink flowers will be produced. So when F1 progenies were crossed, so two pink flowers will be crossed. At that time, when the pure gametes will be means capital R will result into red flower, capital R small r resulted into pink flower, those will be two, and small r resulted into the white flower. So that means there is a, as per Mendel's law, all the flowers should be ratio in the 3:1 by appearance. However, here we get the ratio of 1:2:1 for both genotypically and phenotypically, because there is a variation that happened due to the intermediate phenotype resulted in between. This kind of example is called the incomplete dominance or partial dominance, which is also referred. Yes, yes, Shrey.
So like, is this like blending happening, the mixing?
No. Here, not blending. Here, it's a partial expression. Means both are expressed partially, not completely. No one character is expressed as we are aware that when we mix the palette color, when we mix red with white, it's always pink in color, right? That means there is a dilution factor associated with which resulted into different form. Similar kind of a thing happened in the incomplete dominance. Here, blending is not happening. Here it's the just the partial expression of the both the characteristic.
So like, how is blending different?
Yes, okay. So this is one case. Now we will see the case of the blending, what happened during the blending. So that what we called referred as a codominance. Incomplete dominance is the one thing and codominance is the another thing where the blending is present. So dominance where the offspring shows similarity in both the parents and uh, both parental characteristic will be shown in the main progenial one by blending of the alleles. So here blending happens. So for example, when the red cow is crossed with the white cow, so white is a recessive one and red is a dominant one. We are expecting it to be red one. However, the wrong kind of a cow will be expressed where we can see a white cow with the red patches. This kind of a phenomenon where the both characteristics are expressed due to mixing of which each other by the expressing both the things and the mixing, that is where the blending will be taking place. This kind of a characteristic we refer as a codominance. Okay.
Got it.
Okay, so another classic example of the codominance is a ABO blood group system, which we referred very often. So ABO blood group system is nothing but our red blood cell have the antigens which are present on our red blood cells, which will carry out AB, ABO, different kind of a combination with and without RH factor. So RH factor is also a different kind of an antigen, which is present in some cases, which is absent in some cases. So the way we are say A negative where the rhesus factor is absent, A positive where the rhesus factor or the rhesus antigen is present. So ABO blood group is controlled by gene I. So there is a on the plasma membrane of the red blood cell we have the sugar polymer attached to its surface which controls this activity or the expression of the antigen based on that. So allelic combination that if we can see in this particular chart, so this is allele 1 from parent 1 and allele 2 from parent 2. So if it's IA and IA, we will definitely get the homozygous thing, which will be means both are expressed, which will be expressed as a. If IA, IB, here nothing like that. The A is dominant over a B. It's not like that. The both things will be expressed in the genotypically. That's why we get the blood group AB. Similarly, if one parent has the IA and another just I, means there is a no antigen expressed in A antigen is expressed on that particular part, still this combination will give us the A kind of a blood group. Same happen for the B kind of a system. And if the both parents don't have any kind of a particular antigen which can be present on the red blood cells, this will result into the formation of O type of a blood group that we have the maximum in our population. So this is also example of the codominance. Not it's like A is dominant over B. It's not like that. If the both parents share the different kinds of antigen, they both express by blending together, and that is the very classic example as ABO blood group system. Is this part understood?
Yes, ma'am.
Okay. So, codominance and incomplete dominance are referred as a non-Mendelian inheritance because they don't follow the Mendel's law of inheritance. Now, till these two laws, we have referred the single gene inheritance, that is the monohybrid cross. Mendel also performed some experiment where he uses two different characteristics and see how different kinds of a progeny resulted from those unions. So, in the say for example, in the parental class cross, what he has he choose the yellow kind of a seed with round in shape, and another is a green kind of a seed with wrinkled in nature. So he used the pure form of this. Then the gametes produced during this part will be the identical because this is a homozygous kind of a thing. So the first generation will have the heterozygous yellow and heterozygous round in nature. So appearance wise this is, and uh, genotypically they will be heterozygous in nature. So when it comes to F2 generation, now there are more combinations of the gametes can be possible because now we are referring to the two different characteristics. So four different kinds of gametes will be produced. So capital Y, small r, capital Y, capital R, capital Y, small r, small y, capital R, and small y, small r. Similarly, for the other plant also, this kind of a combination will be there. This kind of a union resulted into 16 different kinds of plants where the all kinds of arrangement, all kinds of phenotypic characteristic that we can see. That means we get the 9 yellow and round kind of a seeds, 3 green and round kind of seeds, then yellow and wrinkled kind of a seed plant 3, and 1 green and wrinkled kind of a seed. So that means that every characteristic is assorted by somehow by the from the gametes, and it can be expressed independent of the each other whatever combination it has. This kind of a law is referred as a law of independent assortment. So, by dihybrid cross, what he has stated, when two pairs of the traits are combined in a hybridization experiment, segregation of the one pair of the characteristic is independent of the other pair of characteristic. So that means two characteristics are not mixing. However, all sorts of combinations are present based on what kind of a gametes are segregated and what kind of a factor they have got over the course of the time. So all the possible combinations that we can get in this way, it's nothing but the law of independent assortment, which is the foundational understanding how the meiosis will be taking place, which we will see later on this part of the explanation. But this is a main law which this is our understanding. This law is help us to understand how meiosis and progenial combinations can be taken place in different organisms by sexual where the sexual reproduction will be taking place. Is this part clear?
Yes, ma'am.
Okay. So Mendel's law, now we are seeing that this is a very crystal clear one, and they are very much visible in whatever we are saying now, understanding the genetic makeup of the organs or how the characteristics are transmitted. However, his work was kept for unrecognized for very long time. He did 7 years of long research and published the data. However, the communication, whatever it has in what we have in nowadays to publish in a journal, in different news, in scientific letters, was not very profound at that time, because we are talking about the era of the 1865. At that time, the publication was not very easily communicated across. Also, what he is referring as a factor is a very discrete idea, means people are not know any idea how it is working, and though it is there, they are not blending. Why this is not happening? This is not very easily understandable and not very much acceptable by the other scientists from that era. Also, his approach is very much mathematical. He put forth various ratios and various understand, because he was mathematician by him own, but putting the biological phenomenon in a mathematical way is very non-acceptable or non-conventional way at his time. And also, the factor what he is referring to is not physically visible, because the discovery of the microscope, high microscope, where we can see the chromosomes and genetic makeup, was not revealed at that time. So this idea was not very much accepted when Mendel stated it. However, his work was rediscovered and republished in by three scientists, De Veris, Corians, and von Skermark. And at that time, Mendel's work was re-recognized, and then he referred as a founder of the modern genetics. So in the 1900, the high power microscopes were under the work, and various we can see the cellular mechanism very clearly by staining procedures, by good optical ways. And at that time, the chromosomes, the nucleus structure, and the chromosomal theory was put forth at that time. So, two scientists play very important role for this. Walter Sutton and Theodore Boeri, they put forth the chromosomal theory of the inheritance, where they see the whatever the current or contemporary knowledge of the chromosomes that we have, which is happening at the cell division, is exactly same what Mendel has stated during his law. And Mendel's law of inheritance will help to understand whatever happening at the chromosomal level, that we can see it now. So this all is revealed, moreover, at the basis of the meiosis process, what happened at the meiosis process, which is involved in the gamete formation. So what they observed that the chromosomes also have the every gene in the paired form. So we have the two allelic combination. So one gene we get from the father's end and another from the mother's end for the same characteristic. So when they are present on the similar kind of a locus, we refer them as a homologous site or homologous chromosomes. During anaphase, we have already learned in the cell division, anaphase of the meiosis one. So we are talking at the first part, where the pair of chromosomes get separated by the firstly they are aligned at the metaphasic plate, and then in the meiosis one the chromosomes get separated, not chromatids, unlike mitosis. So there are different kinds of a combination that can happen at this particular part. So say for example, here we are using the color coding for to understand this process in a better way. So in meiosis 1, say for example, if we see the left side, sorry, it's not- if we see left side of this particular thing, there are two kinds of a chromosomes. One is a orange and yellow, and green and red. So just consider them as a homologous pairs. So when meiosis 1 happens, this kind of a separation can happen by combination 1, which resulted into separation of the chromatids at meiosis 2. Just imagine the other phases are happening in a normal way. So the gametes will be there, where the combination of the orange can be segregated with the green and yellow will be segregated with the red. So which contains the two different kinds of allelic combinations can be happening. Possibility number 2, in the meiosis 1 phase, if orange is get aligned with red and yellow is get aligned with green, there are two kinds of a homologous chromosomes, which will be now we are talking in the two different kinds of a combination. Then the gametes will be resulted into that part will be have orange will be segregated with yellow, orange will be segregated with red, sorry, and yellow will be segregated with the green. So that means there are different kind. What Mendel has stated that the chromosomal uh, what Mendel has observed during his process of the meiosis, or when the two factors, the separation of the allele he has observed, there is a segregational event that is happening of the two factors when it comes to the gamete formation, and they can be independently expressed irrespective of the pair. So there are possible combinations that can happen over the period of the time. So Sutton and Boveri put this thing in a theory of the chromosomal theory of the inheritance, that the pairing and the separation of the pair of the chromosomes will lead to the segregation of the pair of the factors or the genes that they are carrying. So whatever the combination that can happen over the period, by the pairs can be get segregated and the separation can be happen. So this is the similar on the lines what Mendel has observed. So on the basis of knowledge of the chromosomal segregation and Mendelian principle, they put this as a chromosomal theory of the inheritance. Yes,
Ma'am, your orange and yellow are one pair of homologous chromosomes, right?
yes,
And
yes,
blue and red. Green and
yes,
red.
yes.
And so, ma'am, like, why is the green and red pair shorter in size?
No, no, it is just for a demonstration. Just for a demonstration. Nothing else. This is the
Okay,
diagrammatic representation, nothing else, nothing else. Don't consider them they are either shorter or not. This is just for diagrammatic representation.
okay, ma'am.
Okay. So what he has observed, the further experiment, because once the Sutton and Bowery put forth this chromosomal theory of inheritance, there are various experiments were carried out for the where the sexual reproduction and how the genetic characteristic is carried over were revealed by uh, other scientists. So one of the pivotal work for this experimental verification of the chromosomal theory of inheritance was done by Thomas Hunt Morgan and his colleagues. So they discovered the basis of this variation, what happened during the sexual reproduction. So for this particular thing, what model organism Morgan has used is a Drosophila melanoclasta, is nothing but the tiny fruit fly that we can see in our house also, which roam around the fruits. So what is the importance of the using the Drosophila melanoclasta as a model organism? There are four or five characteristics that put this thing to use very easily. Firstly, they can grow on very simple synthetic media, which can be easily reproduced in the laboratory because we have to carry out various experiments. So simpler media is more easy for to feed them or to grow them more easily. Secondly, their life cycle is just for about 2 weeks. So we can get they can complete the different generations. So we can get the enormous data for the by crossing the different kinds of flies during that time, and we don't have to wait for a very long time. So that is another advantage for using this Drosophila as a model organism. Thirdly, they can produce the large number of the progenies, so even we can see the large number of the variations during that time, and that can be used further for crossing. So that is another advantage. Thirdly, the way we have the differentiation in say for humans, that the male and female organisms can be easily separated by their distinguishable characteristic by its appearance also. Similarly, Drosophila male and female can be we can distinguish them very easily. So that way also the experiment can be easily performed. And thirdly, their hereditary characteristic that we want to see can be easily seen under low power of the microscope also. So that can also get everything very handy by the using this organism as a model organism to work in a laboratory. Similarly, genetically also it has some advantages similar as a human. One of the main advantage that the way we have the paired chromosomes, we have 23 pairs of the chromosomes, Drosophila have the 4 pairs of the chromosomes. So they always have the paired form. Similarly, the way our sexual characteristics or the sexual chromosomes are, in case of the female it XX and in case of the male is XY. Similar characteristic we can see in case of the Drosophila, which is the added advantage that we get for to understanding the characteristic or even the human genetics more easily. So here in the diagram also you can see there is a XY chromosome for the male Drosophila and XX chromosome for the female, and the other 3 pairs will be the somatic chromosomes which carry out the different characteristic in them. So this kind of a characteristic will help Morgan to see whatever happening in terms of the inheritance more easily. So, for example, in one experiment, what he did, he crossed the red-eyed female with the white-eyed male. So, two characteristics, and let's say this characteristic is dominated by the X chromosome, associated with the X chromosome. That means the characteristic will be present on the duplicated form, which is a red is a homozygous in case of this particular red-eyed female, and white characteristic is only as or present by the presence of the recessive gene on the one X chromosome that it has for the male. So the eggs will be producing the similar kind, that is RR, and the sperm will have only one X and one Y. So X chromosome will carry out this red color, which is a recessive in nature. So that why it's white in nature. So when the progenies will be produced, they will have different kinds of a combination which will be expressed eventually. As the R is a dominant one characteristic present here, all the progenies, that is the all red-eyed female and all red-eyed male will be produced. Amongst them, if we see the combination, what happens between the one, if red-eyed heterozygous female is crossed with red-eyed male, then what kind of a combination that we are getting for the sperm and also for the egg will be resulted into two red-eyed female, one red-eyed male and one white-eyed male. So that means the by observing such characteristic that it has, this helps for Morgan to put forth that these all the characteristics are controlled by different um, sexual reproduction way, and gametes are helped to carry out this information for further by which is inherited, and the mixing or the by the way of the dominance characteristic observed by the Mendel, similar kind of a variation that we can observe in the progeny in the phenotypic way also. So based on his understanding, there is a one more concept that eventually revealed when we understand the cell biology and how combinations are taking place, is the concept of the linkage and recombination. So linkage is nothing but the two genes of the single pair of the homolog, which there will be the exchange will be happening between two, and they are get linked with each other, and a new combination will be resulted. So there will be the linkage between the two homologous, non-homologous chromatids will be there, which will be happening at the time of the synapse formation during the meiosis, and this will lead to the formation of crossover gametes, which will share some part, and that can help for the recombination and differential expression of the characteristic that we observed in case of the progeny. So Morgan's work is also he also carried out some of the works which is similar like the dihybrid cross what Mendel has done. So he chose the two different characteristics and checked the differential combination resulted in those unions. So what Morgan has observed that he used the two terminology. One is a linkage and another is a recombination. So linkage is nothing but the physical association of the gene on the chromosome. So if we see on a single chromosome, there are multiple genes will be there. Based on the two characteristics that he has studied or whatever, there is a distance in between the two genes, which can also play important role for its expression more effectively in the progenial generation, and also the recombination event, that is the non-parental gene combination, which is not present in the parents but they are present in their progenies, can be also seen which is resulted by the recombination event. So these two terminologies are coined by Thomas Morgan. So what he has done here is a one example that we can see in this particular case. In the parental generation, he used the two characteristic. Say we are talking about Drosophila over here, okay? So in the cross one, what happened in the first cross, the two characteristics will be there, yellow and white, and then there will be the wild kind of a characteristic which is represented by Y plus and W plus. Here what we have to observe eventually, the two genes that we are referring to are present in close vicinity with each other. They are quite nearer in the location to each other. So when he did that cross, the in the F1 generation, he got female with the where the wild type will be there, because wild type is a dominant over the yellow and white type which is present in the parental generation, and yellow and white will be present in case of the male. What he has done, there are gametes will be formed, and in the parental generation, what he has in F2 generation, he found the different kinds of combinations are there. So that can be the wild type or yellow and white, and also these are all present in the two format. One will be the parental format. Parental format means what is present in the parental generation. So that was around 98.7% of the progeny are having this kind of a experiment. Here just consider this diagram as a diagrammatic presentation, because the large progenies are there and finally accumulated data in terms of the percentum. So please don't get confused why this data is represented this way. And similarly, the recombination event is also observed. Here you can see the small the color coded part, that is fall blue part, is get attached with the yellow part. Similarly, this kind of a combination that we can see over here, this kind of a genetic makeup he found in around 1.3% of the population, which is a recombinant type of a population. On the other hand, in the cross B, what he has done, he choose the two kinds of a characteristic, white and the miniature kind of. Miniature is a small kind of a wings that is present in some fly, where the two genes, that is the gene for white color, is located quite far in a distance from the miniature kind of a wings associated with the factor. So here the two genes in the dihybrid cross are located is far from one another on the chromosomal location. So when he did the cross in the F1 generation, white miniature male will be got and wild type will be restored for the female. In the F2 generation, he got the parental generation have 62.8% of the progeny are similar as that of the parental generation. They are either wild type or white and miniature type of a event is there, or they are recombinant type of the event is quite higher, that is 37.2% of the progeny have this kind of a recombinant or new kind of a combination that we can see they are either miniaturized, either white in color, that was varied in number. So what they have observed, that due to the distance present between the two different characteristics, the chances of the recombination will be changed. So the more closer the genes associated with one another, there is a less chance of the recombination that can happen. On the other hand, if the distance between the two associated genes with the similar characteristic is quite far, at that time the recombination event, because of the loose association between the two genes, the chances of the recombination happen at the higher rate. So chances of the recombination happen in this way based on the location of the two two genes associated with the similar kind of a characteristic. So that is what also revealed very easily by the linkage and recombination stated by Morgan's work. After that, yes, yes, Shrey.
And so the two genes are
Hmm.
alleles, right?
Yes, yes. Here we are talking about the alleles, but their location also matter. And here we are talking two different genes with two different alleles. Two different genes means, for
Right.
example, here we are talking about yellow color and white color. Okay. And similarly, the dominant characteristic for the same and a recessive dominant characteristic for another. So that these two genes with different alleles are present on the same uh, close to one another on the complete chromosome thread. Just imagine as a complete chromosome thread, different genes are located over one another. The two genes associated with similar characteristic are present nearer to one another. On the other hand, in case of the cross B, two characteristics are present. The genes associated with the similar characteristic are associated far from each other on the chromosome location.
So ma'am, yeah, yellow and white, which is there,
Mhm.
those both are alleles, right?
Yes, those are alleles, but different genes. Just remember, different genes, different alleles of the different genes.
Ma'am, the gene should be same, right, for the alleles.
Yes, yes, but here, for example, this is one set for the female, okay? So this Y and this Y is one allele. This W and this W is one allele. For the two different genes, yellow is a different gene and white is a different gene. Okay, so the alleles are
Yeah, okay,
present
ma'am,
on the
got it.
this is homologous chromosome. Okay, and when non-sister chromatids are crossing over, at that time we get the event of the recombination. But due to the distance that the nearer in between them, the linkage is quite tighter. So because of that, the recombination event happen in a least way. On the other hand, in case of the cross B, these two genes are present in a distant. So the association between two is quite loose. So at that time, what he has observed, the recombination event occur at the higher rate, which will be 37%. Okay.
So ma'am, this is a dihybrid cross, right?
Yes, this is a dihybrid cross. Yes.
Okay, ma'am, got it.
Okay. So based on this understanding, the further work is also done by Morgan's student. One of them is Alfred Sturtevant, which also used he also helped for calculating the frequency of the recombination by which the gene pair can be get segregated on the same chromosome, and by mapping these all things in a distant way. So the genetic makeup of different organism by mapping in a different position, and what is the frequency of that recombination that can be happen will be revealed by his student. So this work is pretty much foundational work that we now understand how chromosomes are carried out different information in the allelic format, how they share information, how recombination happen, and how we get different progenial traits in the further generation. Okay. So now this is the second part from the chapter of principles and of inheritance and variation. So in the first part, we discuss about how the law of inheritance revealed by Mendel put forth and the theory get proved by when the chromosomal theory of the inheritance was checked, that they are following exactly what Mendel had stated quite centuries ago. So now in the second part, we are now looking at the real life application of how the things are inherited in human beings or even for some other organisms as well. So in the first part, we see that we already are aware that one gene, one protein hypothesis. It is already acceptable. That means one gene is responsible for controlling particular characteristic or particular trait. However, there are some exceptions which are the phenomenal change which can be governed by the multiple genes which govern the single characteristics, which that such inheritance or that traits we usually refer to as a polygenic traits, where a certain characteristic is influenced by the effect of three or more kinds of genes. Along with the genetic factor, here environmental factor can also play a very crucial role. So that can also be the part of the genetic makeup what person has. So one of the classic example for this is a skin color. So we know that we don't always have a black or white. There is so much range that we can see all over the globe that people have different skin tones. So this is one of the classic example of polygenic inheritance or polygenic trait. So what happened in case of the skin color? A, B, C, these three genes control the skin color in human being. So if we put the things in a regular Punnett checkerboard manner, if we see a combination that can happen, that the dominant alleles will be the capital ABC, which we can give the combination will be resulted into the darkest kind of a skin. On the other hand, if the genes are recessive for this trait, that will be the small ABC, the combination will result into the lighter skin tone. Along with that, we are also aware that across the globe we have the different kinds of a sun ray exposure, the temperature variations are there. They can also play important role to have this effect more profoundly. Along with that, we also know that each means as the three genes are controlling single characteristic, the influence of each gene can have the combinations and the effect will be the additive. So for example, across the globe when we can see if say for example this kind of thing, that is the A will be the dominant but BC is the recessive one, is what carried out from one parent, and if it is the A will be the recessive and BC will be the dominant from other parent, that can be resulted into the intermediate kind of a skin tone color. So that's difference we can see across the different races that we have in the world. So this is a kind of a polygenic trait. So each gene will play important role in the addition to get the phenotype to be expressed in a better way. On the other hand, there is another kind of a phenomenon that we call the pleiotropic gene. So when we say pleiotropic gene, here a single gene is responsible for exhibiting multiple phenotypic expression. So that will be the against what our understanding is, that one gene controls only one characteristic. Here in the pleiotropy, what happens? Single gene exhibit multiple phenotypic expression. This happens mostly because the gene is involved in a particular metabolic pathway which is responsible for producing the products, or their effect can be play important role in the various organ systems, or as well as a so that's why the various phenotypes can be expressed in that way, because a single gene is that particular gene is involved in a particular metabolic pathway. So one of the classic example for this is a phenylketonuria disease. So this is a human disease where the mutation occurs in the enzyme coding for phenylalanine hydroxylase. So this is a single gene mutation that can happen in the particular person which has the phenylketonuria. Because of this particular mutation, that can affect the that can cause the mental retardation, also can affect the hair and skin pigment. So we are talking about different body systems that can be get easily affected because of the changes that can happen in phenylalanine hydroxylase enzyme. Similarly, in case of sickle cell anemia disease, in the beta globin chain, the mutation will be happen, and because of this change, we can see that can result into the blindness, that can lead to the liver failure, and that can also lead to the heart. So multiple traits or multiple organ system get affected because of the change happen in a single gene. That is what we call the pleiotropy. So in the polygenic, multiple genes control the single characteristic, and in the pleiotropy, single gene exhibit its phenotypic expression in a multiple way. So then next important part for the inheritance is the sex determination, which is a very important aspect. Now our knowledge of the XY chromosome is quite profound. We know that there are two different kinds of chromosomes which are responsible, that was designated as the sex chromosome. But in 1891, one scientist known as Henking, he was stressing about the spermatogenesis in few insects as a model organism. So at that time, he found that there is some specific nuclear structure present in this insect, which is present in the sperms of the 50% of the population, and 50% of the sperms do not get this kind of a particular nuclear structure. At that time, he was not aware that what kind of a chromosome it is. So he gave them name as the X bodies, which we now, by our current understanding, we call them as a X chromosome. So XY chromosome system is quite profoundly accepted all over the globe for this particular trait. So we know that the sex determination is happen because of the heterogamety. That means two different types of gametes are produced, and that way they can define it will be whether the son or daughter will be resulted from the final union. But there are two kinds of a heterogeneity. When the male is heterogamete, the male can produce the heterogamete. That means two different kinds of gametes are produced from the male. So, however, they can be either will be X chromosome or without X chromosome, which can be designated as X0. And case 2 will be there will be another type of a chromosome system which can be heterogametic. So some gametes will have X chromosome and some will have Y chromosome. So XY chromosome system is what we know about the humans. So human male produces two types of a sex chromosome. One is a X and Y. And in case of the female, it will be the homogametic. That means two X chromosomes will be there. In case of the grasshopper, what happens? It's still male heterogamety, but grasshopper male produces X0. So when we say 0, it will be without any sex chromosome. But it can produce only the X chromosome, and the female will be the homogametic, that will be the XX. So the union will be resulted into this union will be X0 union will be resulted into the male, and XX union will be resulted into the female. On the other hand, in some species, there is a female who are heterogametic, which is we can commonly found in case of the birds. So in case of cock and hen, we can see this kind of a system where two different types of gametes are produced by the female, and male is a homogametic. So in case of, but because if we use the terminology XY again, that will be quite confusing. So that's why we here designated that. So in case of the birds where the female are heterogametic, we name them as a ZW kind of a chromosomes, and in case of the male it will be the ZZ, that is a homogametic. That way the sex determination can be based on the heterogamety and what kind of a union resulted into the development of a zygote or embryo. Is this part clear?
Yes, ma'am.
Okay. So in case of a male, we are aware that we have 23 pairs of the chromosomes. So amongst them are 22 are autosomes, which are responsible for bodily functions, and everything will be in the paired form. And two types of a sex chromosome, one is a X and Y. So XY union resulted into formation of the male child, and XX union resulted into the formation of the female child. This is a very standard system that we can see happen in many species. However, there are some exceptions and some classic cases happen, which is happening in case of the honeybees. So in case of honeybees, the sex determination is happen in the haplodiploid kind of a system. So what happened here? The sex determination happen on the basis of the number of sets of chromosome, how much a individual will get. Based on that, it can be determined, it can be developed further as a male, or it can be developed further as a female. So two types of a developmental process that involved in case of the haplodiploid kind of a sex determination. One we referred as a parthenogenesis, and another word that we use as a fertilization. So in the fertilization process is now we are already aware that the two haploid gametes mate together, and that will develop into the diploid progeny. That is what we call the fertilization process. On the other hand, if development happen without the involvement of the fertilization, that can be resulted in the process is what we call as a parthenogenesis. That means after the process of the gamete formation, it will be immediately developed into the another individual which receives only the half of the number of the chromosomes, that will be the haploid one. So what happens? If you already know the bee system, in the queen is a female, all the worker bees are the female, and males are usually referred as a drone. So which are haploid in nature. So female will produce is diploid, which have the 32 number of the chromosomes in all. So the meiosis process happens in case of the female. Only single female is there in a one honeycomb, that will develop the egg which are haploid in nature. That means they will receive the 16 number of the chromosomes each. Means in each egg there will be 16 number of the chromosomes. If that happens to be fertilized with the male, that male sperm which have the haploid number of the chromosome, that will lead to the formation of the diploid female which have the 32 number of the chromosome. On the other hand, some eggs in the which female queen have laid developed by the process called the parthenogenesis, and that will be resulted into haploid male formation, and that's why they contain only 16 number of the chromosomes. When the sperm formation happen in case of the male or drones, this here the process of meiosis does not takes place. Here the process of mitosis takes place. So that means mitosis is a equational division. That means the progeny and the parental generation will have the exactly same number of the chromosomes. So that means the sperm or the gamete which is formed by the male will have the exactly same number of the chromosome that of the parental one of the male, which will be the 16. So this will be the very interesting phenomenon that happens in case of the honeybees. So the unfertilized egg develop as a male and by the process of the parthenogenesis, and male produces sperms by the process of the mitosis. On the other hand, if the fertilization process takes place, that means the union of the sperm and egg will be developed into the female. So female will be either queen or a worker. Usually worker bees are sterile. That means they cannot produce any further children. Queen is treated only a single bee which is developed diploid female is treated in a such a way that it will be nourished and can be able to have that ability for the reproduction, which is a very special case in case of the honeybees. So the next important part which comes in the influences the process of the inheritance and that can causes the variation is the mutation. So mutation is nothing but it's the alteration in DNA sequence which results into the which can change the genetic makeup as well as the phenotypic expression of that organism. This is a general information that can have. So mutation is another mechanism by which the variation can occur, and also the variation can happen by the phenomenon that we call the recombination. Because in the recombination there is a sharing of the genetic material will resulted into formation of the new kind of the exchange that can happen between the two different non-sister chromatid. Similarly, in case of the mutations, because of the loss of the particular base pair or even a bigger fragment of the DNA or even the gain, that is the addition or the insertion or even the duplication happen by some kind of a error during the replication process, this all resulted into some kind of a abnormality or aberrations. So when we say aberration, aberration is nothing but the deviation from the normal routine that we can call as a aberration. This kind of a phenomenon we normally see in case of the cancer cells. So when we see cancer cell, the programmed cell death is a normal phenomenon that we have. However, cancer cells are immortal in nature. That means the programmed cell death doesn't happen in those cells. So that means normal functionality get deviated due to some kind of a mutation that can happen in case of the cancerous cells. So these kind of a mutations are the result of because of the various factors that are present. There might be chemical, physical. There are some viruses which play important role for the causing different kinds of a mutations that we call the oncolytic viruses. Also, there are some genes present in our body which can get turned on or off by some way, some or other way, which can lead to the formation of some mutations, and that can be lead to the dysfunctionality of cellular system. Such genes we called as the oncogenes. So two categories of the mutations. One is called a point mutation. So when we say point mutation, this mutation happened by the change in a single base pair of the DNA. Even a single base pair can be very much costly for all biological system that can happen inside the body. So one of the classic example is a sickle cell anemia where the single point mutation can cause this disease in humans. Among that also there are two kinds of the point mutations. So one we call that as transversion. So when we say transversion, this is a point mutation where a single purine is get replaced by pyrimidine. So either A will be replaced by T or G will be replaced by C, or it can happen in a vice versa. In short, you have to remember that in the transversion kind of a point mutation, purine is get replaced by pyrimidine or pyrimidine is get replaced by purine. At that time, such mutations we referred as a transversion. Second category will be the transition. When one purine is replaced by another purine or one pyrimidine is replaced by another pyrimidine, this kind of a mutation that we call as transition. So that means if A is replaced by G or T is replaced by C or vice versa, this kind of a cases that can happen in the case of the transition kind of a mutation. So this is a single base pair mutation that we have talked. Another kind of a mutation is called a frame shift mutation. So that means here there is a deletion of the particular chunk of the base pairs of the DNA that can happen, or even the insertion of the newer base pairs of the DNA. So that means we already aware that amino acids that we code usually have the triplets. So the enzyme system read the things in terms of the triplets. So just imagine if there is a deletion of the 2 or 3 codes that can shift the frame completely, and a newer amino acid that can form, and that can be resulted into the ultimate protein effect in total. So that's what we called as a frame shift mutations. Now this is a most important part of this particular topic is a pedigree analysis, and pedigree analysis is a important part to be focused in your IISer aptitude as well. At least one or two questions are definitely there in every year's papers based on the pedigree analysis. So please focus on this part very carefully. So when we say pedigree analysis, this is the analysis of the several generations. So when we know we always do the family tree. So from the grandparents generation, great grandparents to the up to that. So this is a particular trait that is inherited in a particular family that can be get easily analyzed by this kind of a pedigree analysis. Also, this can be used as a good tool to search for a particular trait or particular disease which run in a particular family. So that trait can be traced very easily. Even the eye color, the particular physical features which is associated in a particular family can be easily traced based on the pedigree analysis. So in when we want to put these as a tree format, we cannot use the names as such, or it should be have some general format as it is. So there are some symbols which play important role when we want to do pedigree analysis or draw the pedigree for the particular family. So these symbols are the important. So square will be represented as a male, white square or the hollow square. Circle will be represented as a female. Then if it is a diamond in shape, it is a sex will be unspecified. If it is a filled one, so that can be filled either with dark red color or even black color. It's a filled structure with square, circle or diamond. This will be representing the affected individual. If we want to show mating between the partners, then in between the male and female, we have to draw a line. If this can be happen between the mating happen between the relatives, which is one of the major cause of carrying out the various inherited diseases or hereditary diseases, then we have to show with double line in between. Then if you want to show a particular progeny chart, means the parental generation and a child resulted in those union, then we will show like this kind of a tree. So father, mother, which will be resulted into. So the left one represented the order, means the first child is a female, the second child is a male. Like this we can show the progeny. Even the unaffected parents are, if there means at least superficially, phenotypically they are unaffected, but the union of this resulted into the affected child, we will show like this. The filled square or filled circle will be there, which will be represented as the affected progeny from the non-affected parents. And because if it's a larger number of the progenies that we want to talk about, then 5 unaffected children, say for example, will be shown like in the diamond. They will just write the number 5, which will be 5 unaffected offspring. So these symbols are very important to see the pedigree analysis. So pedigree analysis will help us a lot to trace out the various disorders. So when we say various genetic disorders, we categorize them into the two classes. One is we call the Mendelian disorder and another is a chromosomal disorder. So when we say Mendelian disorder, so the disorder where the law of Mendel's play important role, or we can see visibly the effect of what Mendel has stated in his laws, we can find very easily traced out, or this is a evidential proof that we can see in such kind of a disorders. So they are mainly determined by alterations or mutation in a single gene. So this is one of the main characteristic that Mendelism has, that Mendel has selected the single characteristic and work on that. So this is one of the important characteristic. Secondly, these disorders are transmitted from the in an inheritance fashion. That means the parents will help to have that kind of a mutation to pass through the offsprings or their progeny, which is another important inheritance characteristic of the that Mendel has put forth. Also, the disorders can be either recessive or dominant, which can be easily one or seen when we do the pedigree analysis or chart out the everything. We can see that whether the particular characteristic is dominant or recessive, or they can be either sex linked also. Yes, Shri.
Ma'am, the alteration or mutation are like a frame shift or point mutation.
Sorry. Just a moment. Can you please repeat?
Ma'am, the mutations of the single gene are frame shift or point mutation.
In some cases it can be a frame shift, but that should be occur in a single gene. But in some cases it can be a point mutation as well. But that can the main criteria, it should be happen in a single gene. It should not be controlled by multiple genes.
Okay, ma'am.
Okay. So, so all the Mendel's laws will followed in particular disorder, and we can easily pedigree out the particular trait very easily. So, for some of the classic examples are hemophilia, cystic fibrosis, sickle cell anemia, color blindness. So, we'll see all these things in detail, that what happened in each cases. So, for example, here we can see there is a diagrammatic representation of the pedigree analysis. So this is just a representative one. For each cases, you have to consider all the possibilities. So say for example, if the myotonic dystrophy kind of a disease, which is a autosomal dominant trait, if that is the case, so we can see the parent is already affected and the one unaffected or the carrier is there. In the initial stage, we don't know what kind of a there. Autosomal dominant characteristic, usually if the parental if only a single change in a single chromosome is so powerful that can easily affect the progenies in a irrespective of its sex. So that can happen. So this can happen. We can see in the next F1 generation, we can have the affected as well as unaffected children. Even the affected one can pass, because of the dominant effect of the gene, affected one can pass the disease at least to some of its progeny very effectively. So that is a characteristic of the autosomal dominant trait. In what happened in case of the autosomal recessive trait, this is a case that we can see in the sickle cell anemia. Usually the carriers, which are phenotypically, we cannot see they have some kind of a disease or some kind of a abnormality, but due to the when the recessive gene to be expressed, it should be the gene should be homologous. Because of the carriers can give disease to 50% of their child. The child resulted in those union can have the disease in the future. Similarly, carriers, if they got married, they can also have the disease in the future generation. So this kind of a thing, that is one of the classic questions that they will give you the some kind of a pedigree analysis, and they will ask whether the particular gene is a X-linked recessive, Y-linked dominant, or some kind, what kind of a trait is associated with this kind of a pedigree. This is a classic question that used during the IISc aptitude. So for this, I have particularly included this diagram, so that will help you during your pedigree analysis questions. So there are different kinds of inheritance pattern that we can see. That is, one is a autosomal dominant. So here we are talking about the autosomal chromosomes. So the characteristic of the disease associated with the autosomal dominance is that each affected person usually have an affected parent. So that means, and it occurs in every generation. As you are aware, it is a dominant in nature that can be passed out because of the dominance in even in a single chromosome can be passed very easily to each generation because of its dominant nature. So some of the examples that you can see over here is a Huntington's disease or neurofibromatosis. These are the some of the examples, the disease caused by autosomal dominance. Second class will be the autosomal recessive. So here the classically or general rule is that the both parents are affected person but they are carrier. So superficially or visibly, physically, there is a no abnormalities in them. However, they can pass if they are the carrier. There is a 50 chance of, percent of the chance they can pass disease to the next generation. So autosomal recessive disease is not typically seen in every generation because of its recessive nature. Unless two recessive genes alleles come together and express in a progeny, then and then only the autosomal recessive disease will be passed on further. So sickle cell anemia, phenylketonuria, these are the some of the classic examples of the autosomal recessive disorder. Then there is a X-linked dominant disorder. So when we say X-linked dominance, females are more affected by this particular kind of a thing, and this because of the X-linked, female have XX kind of a chromosome. So females, because of the dominant nature, this can be get more affected very easily, and we can see how it is passing from the same generation. If the mother is affected, then that can pass the disease very easily. So that is a one of the characteristic of the X-linked dominant diseases. On the other hand, X-linked recessive diseases, males are more frequently affected because male children have only one X chromosome. If the carrier mother have the two X chromosomes, so one of the normal chromosome subdued effect of the another X chromosome or diminishes effect of the other, so mother will be the carrier. But if male child resulted in don't union will have the deceased X chromosome, that's why males are more frequent for X-linked recessive diseases, which is commonly we can see in case of the hemophilia. And also, there is a third pattern of the disease we call as the mitochondrial diseases. As you are aware, in the when we learn about the cell system, the mitochondria had its own DNA. And the characteristic of this DNA is that the mitochondrial DNA is always and only carried from the maternal origin. Maternal origin DNA is not involved during the mitochondrial DNA transfer. So if there are some characteristic where the both male and female is affected, but the disease is or the changes had somehow passed in the females only, then we can see that it is nothing to do with the autosomal or sex-linked chromosomes associated disease. It is a mitochondrial kind of effect which can be happen in every generation's child because that is just passing from the mother's generation. So, some of the examples of Kearns Sayre syndrome, these are the examples. Some of the optic neurological diseases are associated with the mitochondrial DNA. Purposefully in this particular diagram, there is a no Y-linked disease is enlisted because Y-linked diseases you can see very easily or can trace out very easily because Y chromosome is present only in the male. So if there is a particular pedigree analysis where you can see only male child is getting affected, we can easily see the affected male is passing out its uh, changes or its mutation to only to the male child. So that way we can easily trace out whether it is a Y-linked disease or Y-linked or it is whether Y-linked diseases or not. So this chart will help you to when you will do actual the pedigree analysis when you solve the actual pedigree analysis question. So let's see some of the examples of the Mendelian disorders and what kind of changes associated with it. So first disease that we want to discuss is the color blindness. So color blindness, this trait is a sex-linked recessive trait. So when we say sex-linked recessive trait, That means it is associated with the X chromosome specially. So X chromosome recessive trait is a, so that means the carrier persons can make the combination of the possible colorblind person. So what is the main symptom of the colorblind person? So there we have the rods and cones which are responsible for having photoreceptors and nervous system, and that can be differentiated into the different color system. So if the person, normally we have the full color vision, but in case of the color blind person, they cannot discriminate between the red and green color, because their cones, their eye system get affected. Because of that, they cannot have the reception for red and green color. All colors are either gray, yellow, blue. In that zone, they can differentiate, but they just cannot differentiate between the red and green, or that can be remain unrecognized. Because of the sex-linked recessive disorder, the defect is mainly found in male, because male receive only one set of the X chromosome, and on the other hand, the female have the double set of the X chromosome. So the one normal X chromosome can diminishes effect of the carrier chromosome, and that way the carrier will have the normal vision, and if only the carrier woman can pass out this particular gene from the affected chromosome, have the 50% chance to get his son color blind. Daughter is normally usually not color blind. This can only happen if a carrier mother get married with the color blind person, then only the two affected X chromosome can be resulted into color blind woman. So which is the rare most cases. That's why we can see, if we say the cases of the color blindness, 8% of the males have the color blindness in them. On the other hand, only 0.4% female have color blindness in them. Second disease is a hemophilia. So hemophilia, normally we know that if we have cut in our body anywhere, the blood oozes out. However, it can be stopped due to the clotting mechanism of our body. In hemophilia, what happens? The clotting mechanism of the affected thing, clotting mechanism get affected, which by which there is a non-stop bleeding happen. So this happens because of there is a single uh, the single protein mutation that can happen in the complete cascade of the event that happen, which are involved in the protein blood clotting mechanism. So hemophilia is also a sex-linked recessive disease which is transmitted from unaffected carrier female to its male progeny. Similar case like that, same as the sickle cell anemia. So this is also a sex-linked recessive trait, which we can normally find that in case of the Queen of the Victorian family, because Queen Victoria was a carrier of hemophilia. Many males in her family we have till now as a hemophilic in nature. Usually females are become hemophilic, means in disease causing way, is very rare, because the female has the least carrier capacity, and the father, means the carrier, should be get married with the deceased one. However, due to this ability, just imagine even a single very small cut can be lead to the enormous bleeding, and so the usually the larger stages of the life won't be achieved for the deceased person. So which is a very commonly in case of the hemophilia. Then third kind of a disease is a sickle cell anemia. So sickle cell anemia, this is an anemic disease. So that means the oxygen carrying capacity of the red blood cell has reduced, which will reduce, develop into the anemic condition. This particular trait is associated with the autosomal linked recessive trait. So now we are not talking about the sex chromosome. So that can happen in any of the offspring. So irrespective of the male or female, that this is controlled by the autosomal linked recessive trait. So here, if the both carriers of the particular disease, but this is a recessive, so both carriers can be resulted into the formation. There is a 50% chance of carrying out this disease genes together, and that can be resulted into the deceased child. So here the changes that can happen is between the two allelic pairs, which is a HbA and HbS. So here we are talking about the hemoglobin or globin molecule in all. So these are the combinations. So the individual which is a homozygous for the recessive trait, that is HbS. So HbS will be have the disease phenotype only. So HbA is a normal and HbS is a diseased one. So heterozygous individual will have the HbA and HbS, which will be the unaffected carrier, and which can pass the disease to 50% of the progeny. So what kind of a disease is there? In case of the normal hemoglobin HbA gene, what happened? It's a polypeptide chain for HbA peptide. On the sixth position, we have the glutamic acid as a in case of the normal protein chain of the HbA gene. So this is a codon for HbA gene. So GAG CTC, which will be resulted into GAG, and which is coding for glutamic acid. Here it is a point mutation that can happen. In case of the HbA gene, this A is changed into the U, and that results into, so GUG is codon for valine. So this single point mutation changes the amino acid itself, and this defect will lead to the low oxygen tension. This is a qualitative loss that can happen, for means the chain is not get abrupted, however the quality change. Instead of glutamic acid, it is replaced by valine because of the sixth position in the beta globin chain. So these mutations lead to the polymerization, which leads to the low oxygen tension that can happen inside the red blood cell. And because of that, the normal red blood cell, which are concave in nature, will be changed into the elongated one, tempered one, and what we call the sickle. Sickle means a more or crescent kind of a shape that we have in case of the moon. So this kind of a shape, elongated shape that we can see, because of that, it can be unable to absorb the oxygen more effectively, and this will lead to the anemic condition for a affected person. That is what we call the sickle cell anemia. Then next disease is a phenylketonuria. So in case of the phenylketonuria, this is also inborn error that can have. This is a metabolic again disorder, which is also the autosomal recessive kind of a trait. So here the affected individual lack the enzyme that converts amino acid phenylalanine into the tyrosine. So phenylalanine hydroxylase is the enzyme responsible for conversion of phenylalanine to tyrosine. So for the affected individual, they lack this enzyme completely in them. So because of that, there is a enormous accumulation of the phenylalanine amino acid into the person's system because of the not conversion, which will be resulted into the mental retardation, because this accumulation can cause the mental retardation for this particular amino acid. Also, they can be excreted from the kidneys also because of the poor absorption capacity of the kidney. They can be easily excreted from the urine for the phenylalanine amino acid. So this is also the autosomal recessive kind of a trait. One more Mendelian disorder is a thalassemia. So thalassemia is also autosomal recessive kind of a trait, which is also associated with the globin molecule. So here is also, when we say autosomal recessive, first thing that you come in your mind, that the unaffected parents are being a carrier, they are passing disease to their children. So here mutation happen in two different chains. What happened? The globin chain, hemoglobin chain is made up of the two chains, that is alpha chain, beta chain. So if the mutation happen in any of these chains in a particular way, that can be associated with the thalassemia. So this causes the abnormal hemoglobin molecule. So that will be the quantitative loss that can happen for this globin chain. So if the changes happen in case of the alpha chain, then it is referred as a alpha thalassemia. So it can produce this particular trait is controlled by the two linked genes which are present on the chromosome number 16. So here two genes control this particular characteristic, that is referred as a HbA1 and HbA2, which are located on chromosome number 16. So if mutation happen in the either of the single of the total 4 genes, when we say 4 genes, because chromosome number 16, we get the homologous chromosome, one from the parent and one from the father and one from the mother. So HbA1 and HbA2 from the father and HbA1 and HbA2 from the mother. So that means there will be the total 4 genes. If the mutation happen in any of these 4 genes, either one or more, that can collectively affect the synthesis activity of the globin gene, and that can be, there will be the less alpha globin molecule can be produced, which can be resulted into the thalassemia. Similarly, if the changes or the mutations happen in case of the beta chain, that is referred as a beta thalassemia. So here beta chain production will be affected. Beta chain production is controlled by the single gene. Like alpha chain is controlled by, the synthesis of the alpha chain is controlled by 2 genes. Here beta chain is controlled by single gene, that is the HBB, which is located on chromosome number 11. So either from the either or parent or in the both parent, its HBB gene get affected, that can be reduce the synthesis of the beta chain of the hemoglobin, and that can be lead to the beta thalassemia kind of a function. So we can see there is some similarity between sickle cell anemia and thalassemia. Both are associated with the globin chain. However, the globin chain genes which are involved in this process are different. This is one characteristic. Secondly, thalassemia is a quantitative problem. That means here the synthesis of the particular chain of the globin, that is alpha or beta, get synthesis itself get affected. That means quantity is reduced somehow. In case of the sickle cell anemia, in case of the globin molecule, only the glutamic acid from the sixth position will be changed to the valine. That way, this is a qualitative change which can have the sickle cell anemia. That kind of a change is a qualitative change. However, thalassemia is a quantitative change. So the effect is quite similar. The red blood cells changes its shape. So in case of the thalassemia, you can see the affected ones have changed its shape, which can again lead to the oxygen absorption capacity of these particular red blood cells, and that can be lead to the anemic condition. Is this part clear? Great,
Yes,
Dani.
ma'am.
Mhm. And now second kind of a disorder is called a chromosomal disorders. So chromosomal disorders, the earlier also we are more talking about the gene kind of a disorder, the single gene or multiple gene or anything the Mendelian laws can be followed. Here, the complete set of the chromosome is present in the additional way or in the reductional way. So here we are talking about the complete absence or excess presence of the chromosome inside the person's body, which can lead to the abnormal arrangement that we usually have the 23 pairs of the chromosome. Just imagine if there is a one additional set of the chromosome for a particular chromosome number, or if there is a reduction in particular chromosome, that can affect the whole body functionality itself. So one such that this kind of a conditions can happen at 2 stages. One stage we referred as the aneuploidy. When we say this, there is a what happened during the segregation of the chromatid process in the cell division. During this process, there is some failure or some cell control mechanism get affected, which lead to gain or loss of the chromosome. So because of the separation during the segregation process of the chromatid, due to loss or gain of the chromosome, the condition that can be affected, that we referred as a aneuploidy. So in case of the Down syndrome, we have the one extra copy of the chromosome number 21. That is the case of the aneuploidy. In case of the Turner syndrome, there is a loss of the one complete set of the X chromosome in human females. So instead of two, they have the Turner syndrome female have only one set of the X chromosome. That is the case of the aneuploidy. Uh, the change can also happen at the stage of the cytokinesis also. So what happened in case of the polyploidy kind of a condition, the failure of the cytokinesis after telophase means complete segregation as has happened, but the after that the failure of the cytokinesis after telophase, the failure of cytokinesis result in the increase of the whole set of the chromosome. So already everything is duplicated, and still the cytokinesis won't happen because of that there is a complete whole new set of chromosomes are present. This kind of a condition is present in case of the plants. When we learn about the seed structure and various kind of the embryo structure that happen in case of the sexual reproduction in flowering plants, we talk about this polyploidy in detail. So the conditions that can be, we can see the more detailed terminology that can be used is one we called if the additional state means we know that all chromosomes happen to be in pair. So there are two for the chromosome number 1. If there is an additional set of the additional copy of the chromosome is there, then we called as a trisomy. This means for example, chromosome 1 have the one more X chromosome, then there are total 3 number of X chromosomes. Such condition be referred as a trisomy. And if there is a lack of any one pair of the chromosome, then we refer it as a monosomy. And this trisomy and monosomy can cause the very serious and severe physiological changes and very serious kind of a disease conditions. So some of the example is a one example is a Down syndrome. So Down syndrome is an example of the trisomy. So trisomies happen in case of the chromosome number 21. If there is an additional copy of the chromosome number 21 is present in some person, the person is affected by Down syndrome. So firstly, this process is described by Langdon Down. So the name is given to honor his discovery. Uh, in this particular, because of the presence of the additional chromosome, there are some kind of a deformities that we can, in case of the even physical characteristic that we can see in case of the child with Down syndrome. So they have the small stature with the round head. Then they have furrowed tongue, which is one of the visible characteristic that we can see. Their palms and even the foot are quite large, and they are quite creased in nature. Their psychomotor or neural activity get hampered very easily. So child with Down syndrome have this kind of abnormalities in them. Then another kind of a chromosomal disease is Klinefelter syndrome. So this particular syndrome, there is an additional copy of the X chromosome is there in case of the male. So male usually have the XY chromosome, but there is an additional copy of the one X chromosome there. So the karyotype means in total we have the 46 chromosomes. In case of Klinefelter syndrome, we have the karyotype with 47 chromosomes. That means XXY kind of a combination that we can see for the male with the Klinefelter syndrome. So overall, physiologically or in the outer way, we can see that the person is male. However, they have some feminine characteristic develop in them. So the breasts are developed for such Klinefelter syndrome person, and usually they are sterile in nature. Means that way they cannot produce the sperms, and that way they are sterile. So their reproductive organs are not very much well developed. This is case for the male. In case of the female, there is a loss of the X chromosome happen in case of the some female, which resulted into the Turner syndrome. So usually the female with Turner syndrome, we have the chromosome number 45. That means they have the X0. One X chromosome is lacking. So that will be resulted into X0 kind of a genetic makeup. So these females are also sterile in nature. So their reproductive ability, because the sex chromosomes, it will get affected. So they are sterile in nature. Their reproductive ability get affected due to rudimentary size. That is a very small or very immature kind of ovaries are there. And also the secondary sexual characteristics that are quite lacking. So instead of they have very broad chest like just like males, this kind of a characteristic we can see in case of the Turner syndrome female.